researchers have successfully identified the crucial component necessary for the self-renewal of blood stem cells, often referred to as the missing piece. Greetings, everyone! Today's video focuses on an exciting breakthrough made by UCLA scientists. They have successfully identified a protein called MICT1 that plays a crucial role in the self-renewal of blood stem cells. This protein helps these cells interpret signals from their environment. The implications of this discovery are significant. It could potentially revolutionize the field of medicine by allowing for the expansion of blood stem cells in a laboratory setting. This, in turn, would make transplants more accessible and pave the way for safer treatments such as gene therapies. One of the key functions of MICT1 is its regulation of endocytosis, which affects how cells perceive signals. In experiments conducted on mouse models, the reintroduction of MICT1 not only restored self-renewal in cells, but also improved their functionality after transplantation. Moving forward, researchers are focused on finding ways to maintain MICT1 expression without relying on viral vectors, as this would be essential for future clinical applications. Without any further delay, let us dive into our discussion. The publication in Nature marks a significant advancement in the field of research, as it brings scientists closer to devising techniques for cultivating blood stem cells in a laboratory setting. This breakthrough has the potential to enhance the availability of life-saving transplants involving these cells and improve the safety of treatments reliant on blood stem cells, including gene therapies. Hematopoietic stem cells, commonly referred to as blood stem cells, possess the remarkable capacity for self-renewal, enabling them to replicate themselves. Additionally, they have the remarkable ability to transform into any type of blood or immune cell that exists within the human body. These incredible cells have been utilized for many years in transplant procedures, serving as a critical and life-saving intervention for individuals battling blood cancers like leukemia, as well as a range of other blood and immune disorders. Nevertheless, there are notable constraints associated with blood stem cell transplants. The task of locating a suitable donor can prove to be challenging, especially for individuals with non-European heritage. Additionally, there may be insufficient quantities of stem cells accessible for transplantation, which can jeopardize the safety and effectiveness of treating a person's ailment. The persistence of these constraints is due to the fact that blood stem cells, once extracted from the body and cultivated in a laboratory setting, rapidly lose their capacity for self-renewal. Despite years of dedicated research, scientists have come tantalizingly close to resolving this issue. Dr. Hanna, a senior author of the recent study and a member of the Eli and Edie the Broad Center of Regenerative Medicine and Stem Cell Research at UCLA, explained that although they have successfully created blood stem cells that closely resemble the real ones in appearance and characteristics, there is still a crucial element missing, as these cells often fail to function properly during transplants. In order to identify the specific element that hinders the complete functionality of these blood stem cell-like cells, Giulia Aguade-Gorgorio, the primary author of the paper and co-corresponding author, examined sequencing data to pinpoint genes that become inactive when blood stem cells are cultured in a laboratory environment. Among these genes, MICT1, which produces a protein with the same name, emerged as crucial for the self-renewal ability of these cells. The researchers discovered that MICT1 controls the process of endocytosis, a crucial mechanism through which blood stem cells absorb signals from their surroundings to determine when to self-renew, differentiate, or remain dormant. Aguade-Gorgorio, an assistant project scientist in the Mikala lab, explained that MICT1 plays a crucial role in regulating the speed and effectiveness with which blood stem cells perceive and process signals. When cells receive a signal, they must internalize and analyze it. However, in the absence of this protein, the signals from the cell's surroundings escalate from subtle whispers to overwhelming screams, causing the cells to experience stress and lose their ability to function properly. Analogous to the sensors found in contemporary automobiles that observe surrounding events and transmit only the most critical data to drivers at the appropriate moment, MICT1 is likened to this technology by the researchers. It assists in making decisions such as determining the optimal time for lane changes or safe turns. In the absence of MICT1, blood stem cells resemble disoriented drivers who, accustomed to depending on these sensors, suddenly feel lost without their guidance. Following that, the scientists employed a viral vector to re-establish MICT1 in order to examine whether its existence could revive the self-renewal capabilities of blood stem cells in a laboratory setting. The reintroduction of MICT1 not only alleviated the stress on the blood stem cells and facilitated their self-renewal in a controlled environment, but also enabled these expanded cells to effectively carry out their functions upon transplantation into mouse models. The team's next objective is to delve into the causes behind the silencing of the MICT1 gene, followed by exploring methods to prevent this silencing without relying on a viral vector, which would ensure a safer approach for clinical applications. Mikola, a professor of molecular, cell and developmental biology in the UCLA College and a member of the UCLA Health Johnson Comprehensive Cancer Center, expressed that preserving the expression of MICT1 in blood stem cells, both in culture and after transplantation, would have far-reaching implications. It would not only enhance the accessibility and efficacy of blood stem cell transplants, but also enhance the safety and affordability of gene therapies that rely on these cells. This breakthrough would pave the way for maximizing the potential of other remarkable advancements in the field. Funding for this project was generously provided by several esteemed organizations, including the National Institutes of Health, the Swiss National Science Foundation, the European Molecular Biology Organization, the UCLA Johnson Cancer Center Foundation, the James B. Pendleton Charitable Trust, the McCarthy Family Foundation, the California Institute for Regenerative Medicine, the UCLA AIDS Institute, the Board of Governors Regenerative Medicine Institute at Cedars-Sinai Medical Center, the Royal Society, the Wellcome Trust, and the UCLA Broad Stem Cell Research Center Stem Cell Training Program. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.